Hello, Eastview Scholars, and welcome to this edition of Sharp Shorts, where we're going to talk about cells. And we'll have a little shout out to Pranav, who has been my buddy, asking me when I was going to drop some more videos. So here we go. Let's drop a video and talk about cells. Okay. And here's what we know. We'll talk about prokaryotes and eukaryotes in a minute, but here's the one thing that all cells have. They have DNA, or some, we should actually just say not DNA, but we should say they have some nuclear reproductive material, DNA. Some prokaryotes are RNA based, but they have some genetic material. They have a plasma membrane surrounding the cell. They have a cytoplasm inside the cell, and then they have ribosomes to make the proteins. And here we go. Two types of cells, prokaryotes and eukaryotes. As you can see by the terminology, meaning before. Karyon is nucleus, so before the nucleus. Evolutionarily, you're looking at the oldest cells, and there's a belief maybe that they were the earliest cells, and then eukaryotes and had some endosymbiotic relationships with mitochondria and chloroplasts and stuff, or early eukaryotes to have membrane-bound organelles. Okay, but these are the oldest. Single cell, no nucleus. In fact, no membrane-bound organelles at all, just a nucleoid region and ribosomes. Eukaryotes, eu meaning new, a number of them are multicellular. You can have, there are single-cell eukaryotic cells, or there are moments when eukaryotes can act in a single-cell manner, but generally you're looking at multicellular organelles. Organisms with membrane-bound organelles. And if we look at the domains that they belong to, domains are the biggest classification, and we break them down into animals, plants, and stuff. The bacteria and archaea are the prokaryotes. And in fact, they used to essentially be one deal. They've split them into two domains now. And then the eukaryotes, where you can see, then they break down into the animals, the plants, the fungi. I'm a fungi. And some of the single cell, basal protists, the flagellates, and these are some of the things that, as when we talk about kingdoms later, it used to be the kingdom Protista was kind of the junk drawer of single cell organisms that weren't prokaryotes, and now they're finding some that are prokaryotic and some that are eukaryotic, although more eukaryotic, and so they're starting to get rid of that kingdom. But that's the area that we would look at. Now, one of the questions we have is, if prokaryotic cells were the earliest known cells and living organisms that were out there, why didn't they just grow larger in the evolution of more complex organisms? And what that comes to, and what you have seen in lab, or will see in lab in some of our work with surface size, surface area, and diffusion. Of course, when we talk transport, most transport occurs in living organisms through diffusion or passive transport, diffusion, osmosis, facilitated diffusion along that line. And it's not a one-to-one ratio. You're really looking at an exponential logarithmic function for something to diffuse into the membrane in a larger one, as opposed to how long it would take to get into smaller membranes. So if you think about that relationship of needing to diffuse the materials into the cell and then have them work, and that it's not a one-to-one ratio but more of an exponential or logarithmic function, then you realize that you could not, the larger you got, that relationship of surface area to volume becomes detrimental to the movement of the materials at a point where you could use it. Okay, so ideally you would have a very high surface area to a lower volume as the ideal, and so therefore having more cells rather than one big cell, but having more cells allows for better use of that ratio and to survive better. Okay. Now here's a quick look at prokaryotes, and like I said, you don't see any real membrane-bound organelles. You see the cytoplasm. There's the nucleoid region. There is the cell plasma membrane. Now this prokaryote happens to have a capsule and a cell wall. That's not a big deal. And maybe have some plasmids. There are some ribosomes in there to make proteins. Where we're going to spend more of our time, though, here is with eukaryotes, both the plant, animals and the plants, which I have side by side there, but we'll look at it here. Here, we're just going to look at the eukaryote, and what you see is some parts that we will run through quickly. Of course, there's the nucleus, which is, in a sense, the control of the cell. It contains the genetic material, the DNA, that is the blueprint for the organism, okay, and bound in this membrane-bound nucleus to protect it. What you see here is the endoplasmic reticulum. There's both rough and smooth, and while attached, they are distinct domains with an intermediate part there. The difference between rough and smooth is ribosomes. Smooth has a different function in detoxification and producing lipids, whereas rough endoplasmic reticulum, when the ribosomes migrate from the cytosol and attach to the side, that's there in the packaging and production of proteins. We've talked about that. Then you have the ribosomes. You have two types. You have free-floating in the cytosol, and if they stay in the cytosol when they're making the proteins, then those proteins are for that cell. And then as they start making the proteins, if they migrate and attach to the endoplasmic reticulum for the rough ER and start making the protein and insert that protein is getting inserted into the endoplasmic reticulum as it's being made, then that's a protein that's going to be packaged and leave the cell to go elsewhere. Okay, and that's the job of the Golgi apparatus, some final packaging and then pinching off these vesicles that because they have the same phospholipid makeup as the plasma membrane, then in a process of exocytosis meld with the plasma membrane and dispense with their materials. Of course, surrounded by the plasma membrane, we'll talk about some of the flagella or microvilli or parts of that later. The mitochondria in both plants and animals is for cellular respiration and producing ATP, the energy. Animal cells have lysosomes, which are, think of them as the custodian of the cell. Anything that comes in through endocytosis or pinocytosis or cleaning up, but they clean up. They have hydrolytic enzymes in them that clean up cell debris, cell waste, anything that comes in, foodstuffs they may break down, etc. There's specialized vacuoles called peroxisomes here, and what they do is essentially they make hydrogen peroxide is what their name is, and that in the breakdown of certain toxins and stuff, which can then be made turned into water. But they're very specialized metabolic compartments in the cell membrane. Here you have a centrosome. In animal cells, there's a centrosome to control mitosis. Of course, the difference between animal and plant cells is no cell wall. And then there's the cytoskeleton that gives support and motility to the cell. What you don't see in animal cells are chloroplasts, which we'll see in the plant cells are there because plants are autotrophs for making their own food from light energy. The central vacuole and the membrane that surrounds it is called the tonoplast, and that's a storage center for plant cells. A cell wall, which we don't have, and plasmodesmata are the junctions that function between plant cells so they can communicate. And that brings us to this question: why all the compartmentalization? Well, you weren't getting bigger; you were getting more cells. Creating more cells makes you more complex, means you have more functions, including communication between an organization, and some of those cells may specialize. So there has to be coordination of the multicellular organism. And in doing that, then as you have to produce energy and you have to communicate and you have to make different proteins and do some stuff, then you need to compartmentalize into those local environments so that if there are things that were incompatible or enzymes that were incompatible, DNA needs to be protected in that nucleus so it's not broken down. That would be somewhat detrimental. Okay. It may be that with that more membrane surface area, you increase the rate of the reactions of things that are going on. And the endosymbiotic theory we'll talk about a little more at a later time, but it is that belief that mitochondria and chloroplasts were incorporated into prokaryotic cells and then became a part of. But initially, were in endosymbiosis, were providing each other a service and a safe environment for the mitochondria and chloroplasts. And we'll dig more into that at a later date. Okay, so here I have the two plant and animal cells side by side. You can see here that the plant cell also has a nucleus, also has the Golgi apparatus and the endoplasmic reticulum. Here's that central storage vacuole, and the tonoplast is the membrane around it. Here is the chloroplast that creates the food, still has a mitochondria, does have peroxisomes to do those specialized functions, does have the plasma membrane. Here are the plasmodesmata that work in between. Does have a centrosome, but does not have a centriole, which when we look at how these two divide and make sure that each cell gets the right amount of DNA, the mitosis is somewhat different and stuff. And no lysosomes, and then no flagella except in some plants that still have sperm for reproduction. But that's just a little comparison to see that essentially most of the cells are the same. Okay. Now, we've talked about most of these parts and what the nucleus does, and certainly you can review in your book what all of these things do. What you couldn't see in these pictures was the cytoskeleton, and it has different size filaments and different things. But what is it for? Well, especially in the animal cell, it's there for structure. It is for structure in the plant, but remember the plant also has a cell wall to give it structure, and then as the plant to work as its skeleton, and as the plant takes in water, that can make it rigid and stuff along that line. The cell wall, the animal cell does not. It only has that plasma membrane, so it needs structure. It needs a skeletal background, and it also needs to anchor all those organelles. Initially, with the microscopes they had back in the day, they were of the belief that all those organelles floated around freely. That is not true, just as it would not be good for your organs to be floating freely within you. It's the same there. So it's not only an anchor, but it's also motility, and some of that motility might be flagella and cilia and some of the things like that, but some of that motility also means moving those vacuoles in a cell, which then can follow essentially the cytoskeleton highway to get out to the plasma membrane and use these motor proteins to essentially walk vacuoles and vesicles and lysosomes and the free-moving stuff along. Now that gets us to how are these things held together? Well, and communicate together. Well, plants are easy. Their cell walls just hold all the different cells together. And we'll see that as a part of the mitosis that we go through and how plants do things differently. Because when they split, they essentially kind of save their cell walls and split new cell wall down the middle. It's not quite that simple, but that's it. And so what they need is they need that communication, and I'll skip back a minute to the plant cells. And to have that communication between cells, then they need the plasmodesmata, which are lined, basically tubes that allow cytoplasm to stream back and forth between the cells to have that communication factor. Animal cells without a cell wall have a few different, as you can see here, 3 different tight junctions, desmosomes, and gap junctions. Now the gap junctions are the communication ones, and you can see they look very much like plasmodesmata. The analogy is that's as close as they are, and that allows cytoplasmic streaming and communication between cells to coordinate all the functions. Now, sometimes the cells need to just be tight and waterproof and prevent leaking and adhere tightly, and that's those tight junctions. That's an overlapping and a kind of a gripping of things to adhere them tightly. Sometimes they just need anchoring, like muscle cells. Don't need a tight junction. This would be more like, say, epithelial cells. But, say, muscle cells need more like these rivets, these desmosomes that anchor, and you can think of them as rivets all along, holding the muscle cells together, but they don't need to adhere tightly. Okay? So anyhow, that is a quick review of cells and their parts and structure and functions and the things that hold them together. Our next video will discuss transport in the plasma membrane and how to that selective permeability of cells allowing things in and out. Until then, scholar on, Eastview.